Okay, folks, let's do the nervous system. And this is going to be a long one. The first check your knowledge is not until more than 40 slides in. So there's a lot to cover here, but let's jump in. All right, we're going to start out by talking about the difference in the central nervous system and the peripheral nervous system, location and function. Location, that's the easy part. If it's the brain, the spinal cord, that's central. If it's away to the periphery, we're talking about towards your arms, your legs, your skin, anything that's moving away from the brain and the spinal cord, all the rest of that is the peripheral nervous system. We'll talk about sensory neurons and interneurons and motor neurons. We're going to talk about voltage gated channels and how they work in action potentials, what myelin sheaths do, both in the oligodendrocytes in the central nervous system and in the Schwann cells in the peripheral. It's the same basic idea. They're kind of like insulation for the, if you think of axons on nerves as carrying a signal down a wire, the myelin sheaths would be the insulation on that wire. Anyway, and then we're going to go over types of neurotransmitters. But let's jump into it. So the nervous system's got two main parts. We got the central nervous system, that's your brain and spinal cord, and your peripheral nervous system, that's nerves that are outside of the central nervous system. Now you can see right away, this is listing things in the central nervous system, and it already has more stuff listed than there. It's a brain and spinal cord. But we've got the cranial nerves coming off the brain, and it's listing spinal nerves that are coming off the spinal cord, the ones that come out through the spaces between the vertebrae. Yeah, definitely cranial nerves central. I will do what the book says. If they would consider spinal nerves to be central, okay, but that seems pretty odd to me because you've got nerves going all the way down to your finger. Anyway, so let's take a look at this. How they work. Your brain and spinal cord are the central nervous system. They're listing everything else as the peripheral, like they said here. I'm good with that. Sensory nerves are also called afferent. The A should remind you that the signal is arriving. It is coming to the central nervous system. The efferent nerves, the E should remind you that the signal is exiting the central nervous system, going away from the brain and spinal cord to the periphery. So if you're sending signals, and that would usually be sensory signals, to the central nervous system, that's afferent. If you're sending signals away, say out to a muscle, that would be efferent. Now, we have other names for them. We call the ones sending signals in sensory, ones sending signals out motor. Great. Understand that sensory don't have to be, oh, there's a bug crawling on your skin. It could be that. But it could also be sending a signal from your stomach to your brain. That's still sensory. Okay. And motor ones, we are sending the signal out to, it could be to the heart, it could be to the adrenal glands, something like that. And the adrenal glands specifically are what they're mentioning down there on the efferent side, where they say there's sympathetic and there's parasympathetic. Sympathetic is the, oh no, there's a bear, I'm in danger. You send a signal, your adrenal gland on your kidneys, the adrenal glands will release adrenaline, also called epinephrine, and that's what causes the fight or flight that they're talking about. That does a whole bunch of things. Same hormone has different effects. It will cause your, say, your muscles will get more oxygen because your blood vessels going there will dilate. But your digestive system, it's going to get less oxygen because the muscles in the arteries and capillaries going to the stomach. They're like, eating right now is not real important. Let's send oxygen to the brain and the muscles so we can get out of here and survive. We'll eat later. So if you release norepinephrine, basically anti-adrenaline, that would be your rest and digest. And both those can be triggered by signals from the nerves. Moving on. All right, so sensory receptors respond to stimuli usually from the outside world, but I already used the example of nerves in the digestive system. But they're going to send the signal to the central nervous system. It will arrive at the central nervous system. It will come to the central nervous system. So it is an afferent signal. Now, integration are neurons that are just in the central nervous system. What that means is that we have sensory afferent nerves that carry the signal to the central nervous system, interneurons that are in the central nervous system, and efferent that carry the signal away from the central nervous system to some target effector. Could be an organ, something like that. Moving on. Okay, here we're seeing regrowth of a nerve, and yeah, it's possible. It's not something that happens real commonly. But the big thing to see right here is if the axon dies out, Schwann cells can grow back and make a new pathway. This is in the peripheral nervous system. We know that because the support cells are called Schwann cells. That insulation is a Schwann cell. If it was the central nervous system, it would be an oligodendrocyte. Same function. They make myelin sheaths. Or I should say, pretty much the same function. Moving on. So we've got two types of cells, the neurons and the neuroglia. The neurons actually carry the signal. The neuroglia, and there's a heck of a lot more of them than there are the neurons. You have support cells that do things like eat invading bacteria. That would be microglia. You have astrocytes, which we'll have to look. I think they're physically hold axons together. Star-shaped, they go up myelin sheath here, but then oligodendrocytes are myelin sheath. I think astrocytes also feed the cells, provide them with nutrients. Anyway, it's easier. In the peripheral nervous system, we got the Schwann cells. Those are our neuroglia. So microglia eat bacteria, debris. Astrocytes provide metabolic and structural support. So yeah, they provide nutrients. Oligodendrocytes are myelin sheaths for the central. Schwann cells are myelin sheaths in the peripheral. How are you going to tell the difference in the oligodendrocytes and the Schwann cells? Actually, really straightforward. Put the things in alphabetical order. The one that's furthest away down the alphabet, I'm talking about the S. So we talk about the astrocytes, and we talk about the oligodendrocytes, and we talk about the microglia. And then the very last thing is the Schwann cell. Well, that's the one that's farthest away from the central nervous system. The Schwann cells are the peripheral. All right, and then we got three types of neurons. We already said we have the sensory ones, the afferent. We have the interneurons that are in the central nervous system, and we have the efferent that send the signal out, away, the motor neurons. All right. Yeah, we've got that. Interneurons are just in the central nervous system. They receive input from sensory neurons, other neurons, sum up signals from others, then communicate with motor neurons. That's not real clear. I'll make it simple. Sometimes you'll get a signal and you won't respond to it. The interneuron might, oh yeah, I noticed the signal from you, but I might need 2 or 3 or 4 different sensory neurons that feed in before I'm like, okay, that's a big enough problem. I've got to deal with it. and then it sends a signal out to the motor neuron. Anyway, 3 parts of a neuron: the cell body, the dendrite, the axon. Actually, you could get more specific. Right where the axon joins the cell body, it's called an axon hillock, and I mean, it could go on and on. At the end of the axon, you could say it's the synaptic bouton and all of that. Yeah, don't worry about it. For our purposes, we got 3 parts: the cell body, which is pretty much like cells that you're used to, and the dendrite, which are little branchy pieces that stick up. They are receivers. Okay, signals coming into the cell are detected by axons. All right, and sorry, detected by dendrites. And the axons, the long part that goes way out there, that axon is the part that sends the nerve signal down to the either the motor, the organ, or the other nerve. That's down an axon. So here we have 3 types. You can see a sensory neuron. Now I want to point something out too, something that hasn't been mentioned yet in the PowerPoints. There are different places where the cell body could be. This has an axon here, and the axon continues up to there. The cell body is in the middle of this axon, okay? And so that's fine. We have receive a signal, and we carry it over to the next cell. We have the end of the axon touching, or coming very close to, actually, not touching, called a synapse, where they get close. But the dendrite gets very close. Sorry, the axon, the end of the axon gets very close to the dendrite. And when it wants to send a signal from this nerve to this nerve, you'll release little chemicals into the space between the two. And that the little chemicals are what we call neurotransmitters. And so what you actually have is electrical chemicals. I'm talking things like sodium, potassium. You have electrically charged chemicals coming down the axon. And when you get here, they will stimulate the release of neurotransmitters, things like acetylcholine, dopamine, whatever, and they will diffuse. They'll come across that little space, and they'll stimulate the next nerve. And the way they do that is they bind to receptors on that nerve, which will open voltage gated will open channels that will then allow sodium to rush in from the outside. And the signal that you had here is now being continued in the next nerve. Anyway, but the point of this slide was to show you that we have a sensory afferent, we have an interneuron, which is short and only in the central nervous system. And you can see it doesn't have a lot of myelination. The myelin cells, the Schwann cells here, we've got a lot less of that going on. And I think that was mentioned. I don't see it here. But there's, when we talk about gray matter, white matter nerves are ones that are heavily myelinated. And the ones that we usually think of, oh, he's really clever, he has a lot of gray matter. Those are the interneurons, the ones that can connect information from coming in and before they go out, that you're considering it or working on it, and oh, what do we do? Those gray matter, they're gray because they don't have all those white oligodendrocyte cells here in the interneuron. Anyway, let's move on. All right, so the myelin sheath covers some of the axons. In the peripheral nervous system, we call them Schwann cells. In the central, we call them oligodendrocytes. The space between the Schwann cells, or I should say between the myelin sheaths, we call nodes of Ranvier. All right, little gaps, and we'll get to this. But if you're sending a signal down the nerve, it's faster to skip from here to here to here to here to just go from node to node to node to node than it is to open channels down the whole length. And yeah, it's faster to do it that way. So saltatory conduction, the conduction goes node to node to node, is faster than normal conduction. All right, so we've talked about that. We've talked about that. Yeah, we've talked about that. Okay, so myelin sheaths made out of either oligodendrocytes in the central or Schwann cells in the peripheral. Those are things that we see on long axons. Short ones don't. Gray matter in the central nervous system is gray because here it is, because it doesn't have axons. White matter is white because it does, has lots of them. Multiple sclerosis, the name means many scars, but it's a disease that attacks the myelin sheath. And if you strip all the insulation off a wire, you're going to have trouble using that wire. Same deal. If you strip all of the myelin off of this long axon, you're going to have trouble sending signals down it. So anyway, the myelin sheath is important in nerve regeneration. If an axon is severed, the myelin sheath can remain and it form a passageway for new fiber growth. That's, I don't know why this is out of order, but there's the picture they were talking about. If the injury doesn't destroy the axon can grow back, and you can, the Schwann cells can grow back, and then you can grow a new axon. Now, I'll have to look this up, but I think, where I was like, nerve regrowth. I think the whole deal here is that we're talking Schwann cells, not oligodendrocytes. Nerve regrowth in the peripheral is probably a heck of a lot easier than central. I mean, we obviously know about like a spinal injury, and then you have paralysis and all. Hold on a sec. I'm going to pause this and check something. Okay, I just checked a journal of physiology. I mean, I just did a search and found a journal of physiology thing that confirmed what I was thinking. The Schwann cells can regrow and do exactly what you see in this picture. The oligodendrocytes in the central nervous system have a lot more trouble. Anyway, let's move on. Still don't know why this slide was out of order, but let's move on. Okay. So physiology of a neuron, we're talking about how we send a signal through the nerve. It's electrochemical. Yeah, it's an electrical signal, but it's caused by little chemicals. I'm talking ions, and specifically sodium and potassium are the big ones. When a nerve is at rest, we're pumping all the sodium out of, say here's our nerve or axon of our nerve. We're pumping all of the sodium out and the potassium in. There's a lot more sodium than potassium. So what that means is, think you have like sodium chloride with positive sodium and negative chloride, but you pump all the sodium out. You're putting all the positives out, well, almost all the positives, and you put a little bit of the potassium in, but you leave all the chloride. So what you're dealing with is a lot more negative in the nerve and a lot more positive outside the nerve at rest. The difference is roughly negative 70 millivolts. And so there they are at rest. You pump things in, and you see the little gates right here. Now, I'll tell you, I don't like, a better way to put it. These drawings are different than I'm used to because they have 2 doors on each channel. And I'm used to seeing a channel with 1 door. So whatever. But the point is, you remember channel proteins are membrane proteins that go through the membrane, and they're like a tube, and stuff can pass through them. We have ones for sodium, we have ones for potassium, but what we also have are trap doors. that can control what can go through that channel, all right? And these doors have little charges on them. So say I'm going to jump ahead. Okay. So first, I guess I should go right here. I want you to see that at rest, we've got all the positive charge on the outside and all the negative on the inside. The signal that's traveling down the nerve is that we're flipping that, we're changing that. So here we've got negative on the outside and positive on the inside. Here we still have the positive on the outside and the negative on the inside. In between those two, you can actually see the positive is rushing in. So what you're looking at right here, oh, okay, that looks more like I'm used to with the one trap door. I was just looking at it wrong. Anyway, what you're seeing right here is the trap door opens and you have the sodium rush into the cell. Okay. So door, we have our channel, we have our door, it opens, and you have a positive charge sodium rushing through. If a positive sodium is rushing in, it brings the positive electrical charge with it. Right? Makes sense? And so that's what makes it more and more and more positive inside. Now, eventually it's going to get so positive that other channels that are potassium channels are going to open up so that the potassium can rush out of the cell. And then before we can use the nerve again, we're going to have to pump things back to where they were supposed to be with the sodium on the outside and the potassium on the inside. So, all right. No big deal. Let's go back and actually read these in order. So at rest, the sodium's on the outside, the potassium's on the inside. The resting potential is negative 70 millivolts. That's cool. And it'll stay that way at rest. You're not going to open those channels until you hit the threshold. Like you could have the amount of the charge to change a little bit, and nothing's going to happen until it rises to the critical threshold. If instead of being negative 70, it's like negative 55, all of a sudden that's so positive inside. I know it's still negative, but it's so close to positive inside that we're going to open the channels. And then it will be positive because whoosh, in comes the sodium, or should go, in comes the sodium. All right. So nerves have to maintain their resting potential and do this. They spend a lot of ATP pumping sodium out and potassium in. Great. And the signal is called an action potential. If you have something stimulate the nerve, activate the nerve, it says, well, a stimulus for pain neurons in the skin would be a sharp prick. Yeah, what's going to happen is that actually will deform the membrane. And that will let sodium leak in. That will take some of those channels and force them open. And then sodium rushes in. But it has to be enough to raise the charge to negative 55 millivolts. That's the threshold. And if you hit that, then you're going to send a signal down the nerve. All right. And these action potential signals are all or nothing. You hit a threshold and you go. If you hit the threshold, you fire. It's not like you fire a little bit or a big amount. That doesn't vary. It's always the same signal. But obviously there are things that are a big stimuli or a little stimuli. So how do you do that? Well, simple. If it's a little stimuli, little stimulus, you fire. This signal goes down the nerve. If it's a big one, you fire, and then you fire, and then you fire, and then you fire, and you don't stop firing. So more action potentials mean it's a bigger stimulus you're responding to. All right, so let's now actually talk about those little gates. By the way, when we're doing the pumping of the sodium and the potassium, where was that? I'm past it. Right there where it says the carrier protein, it's called a sodium potassium pump. It's powered by ATP. When it is pumping the sodium, so this is the top membrane on the cell, and that's the bottom off screen down here. If you're pumping the sodium out and the potassium in, the protein here doing it powered by ATP, that is creating a difference in the charge between the two sides of the membrane. And we call that polarization, okay? Because you're creating poles on two sides of the membrane. So here we have repolarization and depolarization, all right? So depolarization is the stimulus coming down as you open up the gates, and that's where we were. So when an action potential begins, the protein channels open, sodium rushes into the cell. All that sodium coming in makes it positive compared to the outside, and you've got voltage gated channels. They're channel proteins, and they have gates, little trap doors. And what happens is if it gets too positive on the inside of the cell here, more positive, more sodium, sodium's drifting along, it's going to come this channel that's right here. And that channel has a gate on it that is positively charged. And if you get more positive, positives repel each other, it will fold out of the way. But when it does, it just opened the channel, and now lots of sodium comes rushing in. All right. So when an action potential begins, the gates open, sodium rushes in. Adding positive sodium makes it more positive, and the charge changes from negative to positive. Yeah, you can see it. We've got that. Right after and that process is depolarizing. Right after we need to repolarize. And remember I said we have positive sodiums on the outside that are rushing in. But we have positive potassiums on the inside. When it gets super positive, we're going to open the potassium channels. So the potassium is going to rush out. That's going to make a bunch of positive leave the cell. It will return to being negative. But it doesn't mean we can fire again. We're not ready. We've got to put a lot of energy, a lot of ATP into that sodium potassium pump to put things back where they belonged. And then we can fire again. All right, so repolarization, you're pumping things back where they should be. You got it. All right, so here you can see it's tiny fractions of a second, but we're at rest, the signal rises. Oh, we hit threshold. Dang, now the channel's open and in rushes the sodium. And so the charge gets super positive. When it gets positive enough, we're going to open the potassium channels. Potassium's going to rush out. That's going to take us back down to negative. And then we're going to pump until we're back right where we should have been at resting potential. Then we can fire again. So the propagation of the action potential. What we're going to talk about here is conduction down the axon, and they're going to compare axons that don't have myelin sheaths with ones that do. Remember, the ones that do, it's not a complete sheath all the way down, like might have been implied when I said, oh, like insulation. There are gaps in it, those little gaps called node of Ranvier. So say we're talking peripheral nervous system, we got a Schwann cell, we got a little node of Ranvier between, and then on this side of the Schwann cell, we got another node of Ranvier. That conduction down a myelinated sheath is going to be faster than an unmyelinated. So let's talk about the unmyelinated one without the Schwann cells. In unmyelinated, action potential stimulate adjacent parts of the neuron to make action potential. It can be slow because you have to get every part of that axon to do its thing. If it's myelinated, we call, you're jumping between nodes of Ranvier. We call that saltatory conduction. It's way faster, like 10 times faster. Sorry, 100 times faster. And it says the action potentials are self-propagating. Yeah, we got that. That was that all or none. You don't like, oh, the signal goes part way down the nerve, but didn't go down the whole axon. That doesn't happen, because if you build up a charge here, those ions will drift through the axon until they get to the next voltage gated channel section. And they'll be positive enough that the trap door will swing out and more sodium rushes in. So if you've got enough to start the signal, you'll have enough to start the signal here. And that one will create enough to start the signal at the next spot, which will make enough to start the signal at the next spot. That's why it goes down, it's self-propagating. That wouldn't be true if we couldn't pump our sodiums and potassiums to where they need to be at rest. All right, so here we have a picture of the action potential. You can see that the signal is happening here because instead of the inside of the axon being negative and the outside being positive, we've had sodium rush in. Well, that's enough to open the gates, and now we have the potential here, and we're pumping things back there. But that's enough that we're about to open the gates here, et cetera, et cetera. As long as you got the ions. All right, the refractory period is the time when you can't fire the nerve again. I know, like, I'm moving my finger as fast as I can, and it doesn't seem like there's much of a gap. There's a gap. There is a time when you cannot fire again. You've got to pump those ions back. I guess what I'm saying is your nerve can only go so fast. You can't go faster than you can pump things where they should be. And that's good, because that means that your signal can only go that way. It can't come back the wrong direction. If you think about it, like, okay, I sent the signal down the axon and refractory period, I can't do anything. That means that when this node, say, we open our gates, our sodium rushes in, diffuses along, it's going to open up the gates at the next area. Well, now we're in refractory period. We can't have this one that opens up over here, the second one. We can't have its ions drift back and mess with this one. No, that doesn't happen because we're in refractory period. You may have ions drift over, but they're not going to do anything. All right. So the end of the axon, we already said that's the synapse. So here we have a nerve, and it comes down, and the end, the semi arms the axon. It ends with a bunch of little branches, and those branches end in axon terminals, which I think are what we used to call synaptic boutons. Hold on. Gotta check that. Yep, turns out that a synaptic bouton is just an older way of saying axon terminal. I kind of like it. It sounds super nerdy and sophisticated, but oh well, work with it. We're going to use the modern term. So these axon terminals, think my fingertips. All right. We have an axon, it branches. We have the axon terminals. It's the terminal because it's the terminal in the very end. They come very close to the dendrites of the next cell, or say this is a muscle fiber, very close to the muscle fiber. They don't actually come right up to it and touch it. There's a tiny, tiny little space. And in that space, they call that little space the synapse or synapse. And into that synapse, you're going to spill little chemicals, neurotransmitters, that will transmit the signal from this nerve to the next nerve, or this nerve to the muscle. I'm talking about things like acetylcholine is the famous one, or dopamine. I mean, there are lots of them. But we're going to spill chemicals into that synapse. You're going to diffuse across to the target. Could be some organ, some muscle, probably another nerve, actually. And they will bind to receptors. Those little neurotransmitter chemicals will bind to receptors on the target, and they will open, sounds familiar, channels through the membrane that let ions rush in. and you continue your signal. All right, so let's do this. So the synaptic cleft is the little gap, and we're going to spill neurotransmitters into it. They are stored in little vesicles that are in the synaptic terminal. I'll say that in old speak. You have little vesicles full of neurotransmitters that are in the synaptic bouton, and they will, when you stimulate and try to send that signal across the synapse, they will, the vesicles will bind to the plasma membrane and open up, become part of the plasma membrane, and what they had inside is now outside of the nerve, and it's diffusing across the synapse to the target. And that target that has receptor proteins on its membrane that will receive the acetylcholine or whatever the neurotransmitter is. And those targets, whenever the neurotransmitter binds, you open up a channel and ions rush in. All right. So nerve signal, this is how the synapse, how it works. Nerve signal goes down the axon, it gets to the axon terminal. Calcium ions rush into the terminal. They cause the vesicles to merge with the plasma membrane of the nerve. The vesicles spill out their neurotransmitter into the synaptic cleft, the gap, and it diffuses across the cleft to the receiving membrane of the target cell, whatever the target cell is, and they'll bind to receptor proteins. You can see it happening here. When they bind to, give me my mouse, thank you. When they bind to this receptor protein, it will open a channel, and now you can have chemicals rush in. All right, so you got it. Depending on the type of neurotransmitter, the response you get could be exciting or inhibiting. Like, what the heck with the inhibiting? Well, suppose you're an interneuron and you're getting signals from this sensory neuron and this sensory neuron and this sensory neuron. It's very possible to have this one says do something and this one says, no, not yet. No. And in order to say, oh no, not yet, you would send a neurotransmitter that makes it harder for this one to be listened to. Maybe this receptor, if you don't have something else, it will cause a channel to open. But this neurotransmitter, it will bind to that same channel and plug up the works so you can't have, so it won't open, it will ignore the other neurotransmitter, that type of thing. And the example they're using right here, and it's a good one, is if you bind to the channel that has potassium leave, then it's going to not just be polarized to -70, it'll be hyperpolarized. The charge will go down because the sodium, the potassium leaves. And so then you would need 2, 3, 4, 5, you would need multiple signals to raise it up to the threshold before you fire. All right, moving on. So we filled this cleft with neurotransmitters. How do we get rid of them? We're going to have enzymes that break them down. That's one method. We could have the receiving cell actually pull them out of the cleft and reabsorb them. But you could have something like acetylcholinesterase. Remember, if it ends in ACE, it's probably an enzyme. Acetylcholinesterase breaks down acetylcholine. So it's like it was never there. All right. You don't want to have neurotransmitters there for a long time. Prevents continuous stimulation of receiving membranes. The reason I'm pausing here, I wanted to jump in and say, but that's what the drugs do. But there's a whole section at the end of the chapter about drugs. So we'll talk about that. If a neurotransmitter was left there in the cleft, you wouldn't be able to respond to new signals properly. Anyway, so neurotransmitter molecules. There's the acetylcholine I was talking about. It's one of like 100 neurotransmitters, everything from norepinephrine, epinephrine, the adrenaline, dopamine, serotonin, you feel good and relaxed. All right. Anyway, they're neurotransmitters. They're lots of chemicals. You have synapses between nerve and muscle, nerve and organ, nerve and gland. All of those use different types of neurotransmitters. Acetylcholine and norepinephrine is super famous because they work in both the central nervous system and peripheral nervous system. What that means is they were probably detected pretty early, and there's been a lot of research on them. In the peripheral nervous system, they act at neuromuscular junctions. Where I was talking about how you have a motor unit, say this is a muscle fiber, and you have the axon coming in with the synaptic butons getting close. That little cleft right there that you're going to put acetylcholine into, that's what will make this muscle fiber fire. That's how the signal gets from the nerve to the muscle. All right, moving on. In the peripheral nervous system, acetylcholine excites skeletal muscle, but it inhibits cardiac muscle. Just accept it as it is. It's weird, but okay. Norepinephrine excites smooth muscle. Okay. In the central nervous system, norepinephrine is important for waking, dreaming. So I'm hoping they have a nice table here for us. And of course they don't. So let's just go with, yeah, they've only got 3. They've got 3 different neurotransmitters here that are harping on. So just have to memorize what acetylcholine does, what norepinephrine does, what serotonin does, and what they do in the central and the peripheral. The actions can be different. Know that. Anyway, oh, and there's the thing about drugs that I was talking about at the bottom of this slide. Many drugs act at the synapse, and some interfere with neurotransmitters. Like I said, there's a whole section on different illicit recreational pharmaceuticals at the end of the chapter, and we'll deal with that a lot more when we get there. But let's move on. Okay, so yeah, okay, we're close. So synaptic integration, remember interneurons? Yeah. Integration is summing up multiple signals are coming in. Some are excitatory, some are inhibitory. If a neuron gets enough excitatory signals, so many exciting ones that they're way past the inhibitory ones, it will fire and send off the axon. On the other hand, if it's getting more inhibitory and not as many excitatory, it may get hyperpolarized as you have all the potassiums leave or something. But for whatever reason, whatever the mechanism, it's not going to fire. So here we have a picture. This is an electron micrograph, and you can see the actual synaptic butons or axon terminal, how they're getting right close to this cell, and they can put signals, they can spill neurotransmitters into the little gap, the little cleft between the first neuron and the second one. Anyway, let's move on. So here we have excitatory and inhibitory signals. Remember, our resting potential is -70 millivolts. If you have signals coming in that are excitatory, they get you closer and closer and closer to the threshold. Remember, once we hit that threshold, we're going to fire. And it's an all or nothing. We send the signal on full strength. So if we just barely have enough stimulus to get to the threshold, we fire. If we have way more stimulus than we need to get to the threshold, we fire. It's the same way. We tell the difference between the two and how often we fire. Anyway, if you have inhibitory signals, you have your potassiums leaving the cell and you're getting more and more and more negative, and then it takes a lot more stimulus to get you to the threshold. All right, there's the check your progress. We made it. Okay, so that's the first one. There are 5 of these things. I told you this is a long PowerPoint. I'm going to pause and you guys can check yourself and we'll continue in a little bit. Okay, I'm back. This is a really huge chapter. And I just made it a little longer. I modified the PowerPoint, but I did it by adding pictures at every step so you don't have a wall of text and then, oh, what was that thing? And then I scroll forward, scroll back. I think we'll be able to do this a little faster. But this next section, 14.2, we are going to do the anatomy of the central nervous system. I'm talking the whole spinal cord and almost all the brain. It's a lot, and this is just scratching the surface. But oh my! So let's jump into it. Learning outcomes. You got to learn the structures of the spinal cord and what their functions are. Structures of the brain, function of each. Lobes in major areas, distinguish between primary motor and primary somatosensory areas of the brain. That there's actually a sulcus, a groove that separates the motor ones are in front and the sensory ones are in the back, but we'll get into that. All right, so central nervous system's the brain and the spinal cord. You know that, you've heard that. That'll work a little better. The brain and the spinal cord are both surrounded by bone. We're talking about your vertebrae and your skull. Between the nervous system and the bone, you have protective coatings called meninges, like around the brain. One of these is the tough outer one would be dura mater. Think of durable. And the one below that is barely there, thin like spider web, the arachnoid mater. Yeah, like spider web. And then the one that's in intimate contact with the grooves, the gyrae and the sticky epi parts of gyrae and the grooves down the sulci. That part in intimate contact is the pia mater. And between these layers, you have little bits of cerebrospinal fluid. And also inside the brain, you generate the cerebrospinal fluid inside ventricles, spaces in the brain. And there's actually a canal that goes all the way down the spinal cord that has the cerebrospinal fluid in it. But we'll talk about that. Anyway, the meninges are these membranes that are wrapped around the nervous system, the central nervous system anyway. And if you get inflammation of that, it's meningitis. There's bacterial meningitis, there's viral meningitis. It's all bad. Moving on. Hold on just a second. Okay, so the central nervous system, brain, and spinal cord. You know that this receives information, it initiates control. If you remember, interneurons are only I'm talking gray matter, are only a central nervous system thing. Let's move on. And here we go. All right, so the cerebrospinal fluid is found between the meninges. It cushions and protects the central nervous system. When someone does a spinal tap, turn it up to 11. Sorry, bad band joke. When someone does a spinal tap, a lumbar puncture, they're drawing out, they're taking a syringe and sucking out cerebrospinal fluid to test it. The cerebrospinal fluid is in the 4 ventricles of the brain. You got the 2 lateral. You got one in the middle they call the 3rd ventricle. And then you have one in the cerebellum, little triangle shaped one they call the 4th ventricle. Anyway, those ventricles, they're all connected by little canals, and that's what makes the cerebrospinal fluid. You also have the, after the 4th ventricle, it continues all the way down the spinal cord as a central canal. Anyway, so excess cerebrospinal fluid is usually drained away into the cardiovascular system. If you get a block and you're an infant, the brain, because the skull is still soft, the brain can enlarge, push on the skull, and cause this huge head they call hydrocephaly. if you're an adult, your brain, your skull can't swell to accommodate the brain. So the brain gets pushed up against the skull and it can cause brain damage and can kill you. So no blockage is wanted in cerebrospinal fluid. Bad. All right, so there are ventricles. You can see the two lateral, and I'm going to move my mouse over them. The two lateral right here, and then that very thin third ventricle that connects them. And below that, the fourth ventricle here. All right, so we've got those. The central nervous system is made out of two types of tissue, gray matter, white matter. Gray matter is short axon, neurons that have short axons without myelin sheaths. And white matter, they tend to be longer axons. They're collected into bundles that we call tracts. And those long axons, they got insulation, they got myelin sheaths. All right, and since this is central nervous system we're talking about oligodendrocyte sheaths. We're not talking about Schwann cells. Schwann cells are to the periphery, the peripheral nervous system. So the spinal cord goes from the base of the brain through the foramen magnum, the big hole. That's literally what the name means. It's the hole at the back of the skull. The spinal cord comes out of that and travels down in the vertebral canal through the vertebrae. Structure of the spinal cord, we're going to do a cross-sectional picture. It's that one right there. We'll talk about in a second. I want you to know I hate this picture. There's no reason in the world why the diagram and the photograph should not be oriented in the same direction. They should have flipped that upper picture so it would match, but oh well. Anyway, cross-section of spinal cord shows that central canal full of cerebrospinal fluid, gray matter, white matter. The gray matter's to the middle, the white matter is around it. The gray matter kind of looks like a messed up butterfly. Spinal nerves go into the spine. They come from the spinal cord through intervertebral foramina, the little spaces between the parts of the vertebrae. And in between those vertebrae, you have the intervertebral discs, which are made of fibrocartilage. A ruptured disc is super painful because then you have two vertebrae coming together and pinching the nerve that comes out of that foramina. So, not good. All right. So, if you look at this picture, the dorsal, so imagine our messed up butterfly, The narrower, more vertical parts are the dorsal parts. And here in this photo at the top, they were oriented it upside down. So that's annoying. But still true. White matter to the outside, gray matter to the inside. There's a central canal. And you can see how it lays there. Also, you can see that coming out of the foramina, we have these spinal nerves, and we have a dorsal spinal nerve and a ventral spinal nerve. They come together to form these nerve tracts. The dorsal one is going to be bringing in sensory information, and the ventral one will be sending out motor information. But once you get past this ganglia where the cell bodies are, they're all hooked together and they run together as one nerve to whatever part of the body they're going to. All right. So, oh, and it was right here on the next slide. I could have said that. So gray matter is centrally located. The dorsal root of the spinal nerve contains sensory fibers entering the gray matter. So dorsal is sensory, ventral is motor. The dorsal and ventral roots join and they form a mixed nerve. Spinal nerves are part of the peripheral nervous system. Okay, this makes me much happier. I'm not going to jump back to that initial slide of the central nervous system at the beginning of this video that showed the spinal as well. I was like, I don't think so. Well, the book doesn't think so either. It's just a messed up diagram, which doesn't surprise me anymore because messed up diagram. Moving on. All right. So here we are. And you can see that the gray matter is the butterfly. That's unmyelinated nerves. The white matter, which is to the outside, that's myelinated nerves. These myelin sheaths are made out of oligodendrocytes, not out of Schwann cells. All right, so let's move on. The white matter surrounds the gray. The white matter has ascending tracts that go from body up to brain, and descending tracts that go from brain down to body, then as you're down the spinal cord, and then when you're at the level where you can go out to the spinal nerve where you need to go, you go out the intervertebral foramina. Okay, good. The spinal cord is a gateway for pain signals. Motor signals from the brain go down the spinal cord out to the muscles. Let's talk about paralysis, specifically paraplegia and quadriplegia. If you damage your spinal cord in the thorax, you're going to probably have quadriplegia, where the lower body, lower legs are paralyzed. Damage in the neck, which is higher, that'll be quadriplegia. That'll do that. That will limit or it will impair the use of all four limbs. We have that, and just a reminder, what we were talking about before. Damaged nerves in the peripheral nervous system. You can have Schwann cells form a sheath and grow a new axon. Oligodendrocytes don't do that very well. So that's why spinal cord injuries, they stay with us. All right. Now, the functions of the spinal cord, reflex arcs, lots and lots of reflex arcs. You get a stimulus that sends a signal to the spinal cord through the dorsal, the afferent, and the sensory, and interneurons that are in the central nervous system will integrate it and send signals to the motor neurons. The motor neurons are going to send signals to the skeletal muscles to make them contract. An example of that, a reflex. Imagine if you touch a hot stove and you don't need to detect the heat and the pain. Your body will already be sending signals, like that signal gets to the nervous system, gets to the central nervous system, and yeah, you'll use the ascending tracts to go and tell the brain about it. But before that even happens, the spinal cord is like, no, get your hand off of that, and sends a signal to the motor neurons to pull the hand away. And that's why you can pull your hand away and then you feel the pain. Okay, so let's move on. You can also do reflex arcs for internal organs. If your blood pressure falls, you have pressure receptors in the carotid artery and the aorta, and they're going to tell the brain to tell the heart to pump harder so that you can increase blood pressure. All right, now we're at the big scary part. We got to talk about the parts of the brain. The cerebrum, like cerebral cortex, like where everybody talks about intelligence, but the cerebrum or telencephalon, the diencephalon, the diencephalon, cerebellum, and then the brainstem below that. But there's a pretty picture here. You're going to see this picture many times. But when we say the cerebrum, that's the main part of the brain. We're talking about this. Okay. Then when we say the diencephalon, we're talking about this. That's what it's trying to draw, is that you're getting up under the brain. Trying to show you. So diencephalon, the cerebellum right here, and the brainstem right here. We'll be going over all of those with what I hope are better illustrations. I've been screwing with the illustrations to try and like circle, I'm looking at this thing, or we're talking about this thing. Anyway, so those are the four main parts, and the telencephalon or cerebrum is the biggest part. This is, we send sensory information in there and do integration before we do voluntary motor response. and it communicates with the other parts of the brain. That's great. Okay, if you look right here, there is that we have the two lobes in this model of the brain. Okay, well, left side, right side. I shouldn't call them lobes because we're about to go over lobes. But right here, there's a huge groove. It wiggles, but it goes all the way down. Okay, that's important because we're going to talk about how you have motor control in front of that groove and sensory integration behind it, but we'll get there. All right, so the longitudinal fissure is the deep one right here that divides the left and the right hemispheres. And they are connected through the corpus callosum. That's this area right here, which I've heard some people describe that as this whole area as looking like a chicken. Think this is the bird's head and beak, and then it has the comb on its head right there. So we've got our chicken with the comb on top. That's the corpus callosum. Those are tracts that connect the left hemisphere and the right hemisphere and let them talk to each other. The little cherry in the beak of that bird, that's your pituitary. The chiasma is right there, that's the eye. Anyway, we'll get to it. We'll get to it. So the bird's head itself is the thalamus and hypothalamus. Anyway, yeah, let's move on. Let's move on. Okay, so that corpus callosum that connects the two halves, that is a tracks going back and forth between the hemispheres. The gyri are the thick folds, the sulci are the shallow grooves. Moving on. So the hemispheres are further divided into lobes. We have a left hemisphere, right hemisphere, but they've got frontal lobes. Now, you'll notice these names are the same as the bones because they're divided up similarly to overlying bones of the skull. So we got frontal lobes, we got parietal lobes, we got occipital lobes, and we have temporal lobes. We'll do it again. Frontal lobes, parietal lobes, temporal lobes, occipital lobes. All right, and here's the pretty picture. You're going to see this picture with modification again and again and again. Moving on. So the cerebral cortex, the gray matter, it's thin. It's got a lot of grooves and wrinkles in it. The thick, wide areas that are raised are called gyri. The little invaginations that go down in, those are called sulci. Anyway, but the gray matter in the brain, you remember the spinal cord? We had the white matter on the outside and the gray matter on the inside. Think opposite in the brain. The cerebral cortex is where we do sensation, control our skeletal muscles, thought, all that jazz. Moving on. So the primary motor area is in the frontal lobe before the central sulcus. I know that's like, huh? So we're talking here. Well, that's the association area, but same idea. It's right here, primary motor area in blue, okay? So that groove on the side that comes down this area just in front of that. And after that, we'll talk about the area in back of it, which is sensory. That's the part they have in pink. So we're going over the blue stuff now, the stuff in front of the groove. The primary motor area, voluntary signals to skeletal muscles. There are large amounts of muscles for swallowing, facial expressions, things like that, because that's important for understanding if the person talking to you is threatening or scary. It's a good part of how we function as a social species. So find control of all of our muscles that can do emotion, that convey emotion. If you get a lack of oxygen to the primary motor area of the brain when you're during birth, like a kinked spinal cord, sorry, a kinked umbilical cord, you could end up with cerebral palsy, weakness of the arms and legs. All right, so that's the blue area. And notice that this little tiny bit controls muscles of the leg. And this huge portion here controls muscles of the face. And an area, again, much bigger than the leg, controls salivation, chewing, and talking. So the muscles that need fine control, it's large portions. Anyway. All right, the primary somatosensory area, that's the pink area on that last diagram, it's just behind the central sulcus. Information from skeletal muscles, information from skin. We're looking at senses. Again, hands and face have huge amounts of space, especially your hands, your fingers, your thumb. Super important to how we live as primates, our opposable thumb. Lot of nerve control going on there. So a lot, I should say, a lot of sensory information processed there. All right. Now let's go over reception areas. Taste in the parietal lobe, visual in the occipital lobe, auditory areas in the temporal lobe. Makes sense because you remember that's where the external auditory meatus, the hole going into the ear canal, is. Olfaction at the frontal lobe. You might think, what? Well, but it not only is it close to the brain, the bottom of, remember your skeletal muscle, your bones of the skull, the ethmoid bone that forms the ethmoid, it has the ethmoid sinus at the back and all that. The cribriform plate of the ethmoid bone, it's all perforated and has holes in it like a colander, and it has sticking up in the middle. So we have holes where scent can rise up to the olfactory nerves, olfactory bulbs, holes where scents can rise up, and separating the two olfactory bulbs, we have the crista galli, the sail that sticks up, think like sailboat. Anyway, so it makes sense that that stuff would be processed in the frontal lobe because that's what it's closest to. Anyway, okay, so the association areas, the premotor area, that's what we just talked about. That's control and association of things that involve skeletal muscles. The somatosensory area, that's processing sensory information from muscles, nerves, bones. Sorry, why did I say bones? Muscles, skin. And again, you already saw that the amount dedicated to processing information from the nerves in the hands is a huge, huge part of it. Other association areas, the visual. Remember we said visual is occipital. It's not the very back of the occipital. The association area is just in front of that. So recognition of people's faces, things like that, that's where that is done. All right, now auditory association, that's on the side right next to the ears, which again, it makes sense. Okay, but that's auditory association. It's part of the temporal lobe. The prefrontal area right there, it's part of the frontal lobe, but on each side, not right at the front. That's reasoning, planning, critical thinking. Wernicke's area, which is back of your left side temporal lobe, understanding of words. And that's both things you hear and things you see written. That's super important. And it's the left temporal lobe, not both. So, moving on. All right, now Broca's area. Remember we had the motor association area just in front of that big groove? In front of that, near the bottom, is Broca's area. That's left frontal lobe. And that's muscles for speaking and writing, fine control. Muscles for speaking. So understanding words, that's on the left side. Writing words and saying words, also on the left side. Language. So Broca's area. Now there's the white matter in the middle. Remember we said that spinal cord has white matter to the outside and gray matter in the middle. Well, in the brain, it's the opposite. You got the gray matter of your cerebral cortex on the outside, and the white matter is in the middle of the brain. Okay. It's white because it's myelinated. Now this is central nervous system, so that's going to be oligodendrocytes. It's not. Oh, what? Okay, so it's central, so it's going to be the oligodendrocytes that form the myelin sheaths, not the Schwann cells. The Schwann cells form the myelin sheaths in the peripheral. Sorry about that. Brain fart took me a second. Anyway, the descending tracts from the primary motor area communicate with lower brain centers. Ascending tracts from lower brain centers send sensory information to primary somatosensory. Basically, in the brain, coming from the spinal cord, we've got sensory up, we've got motor info down after we've integrated and made decisions about what motor things need to be done. So these tracts of white matter also take information to and from the sensory association areas, motor association areas that we've just been talking about. You also have white matter connecting the two hemispheres of the brain. On the corpus callosum, there's where's a good picture? There we go. If you think of the corpus callosum as being like a bird with the pituitary being like a cherry in its bill, make it a bird that's a chicken and it has a comb on top. This comb that's on top, I'm talking about this crest area right here. That's your corpus callosum. That's nerves going back and forth between the two hemispheres, letting the hemispheres talk to each other. I probably should have used like that, would have been easier to see, but you get the idea. All right, let's move back here. So the basal nuclei are masses of gray matter deep within the white matter. So we actually have gray matter, white matter, gray matter. All right. These will take commands, what we should do with muscles, and integrate them to make choices about the right muscle groups. Parkinson's disease, where someone has trouble controlling their hands and they get the shakes. That is caused by degeneration of some of the neurons in these basal nuclei. One of my old profs, the guy who taught me to use an electron microscope, he had Parkinson's, and you could tell it was really frustrating for him because he would try to do something like gluing a specimen to a stage, and his hand would shake too much to do it, and he would sigh, and he would say, you'll have to do it for me, and we'd do the thing. It's really disheartening because they're slowly losing more and more control of their body, but they're mentally still fully there. Anyway, moving on. So let's, okay, here we go. We're about to talk about the diencephalon. Everything we talked about before here was telencephalon. Sorry. Or, yeah, there we go. spinal cord, the brain, the four parts. Everything we've talked about so far in the brain is the cerebrum, which is the outer and the main portion. I'm talking, it's all part of this, okay? You might say, oh, there were some things down there. Yeah, but they were down around. Okay, so it's the outer portion. That was the telencephalon or cerebrum, cerebral cortex. We are now going to talk about the diencephalon, which is in the middle around that third ventricle. The ventricle, this like the cheeks of the bird. If this is your bird, the cheek, that's a hole, that's a space. That's the third ventricle that fills with cerebrospinal fluid, that makes cerebrospinal fluid, okay? And the thalamus would be like the beak, and the hypothalamus would be actually, sorry, the hypothalamus would be like the beak, and the thalamus would be above that. That'd be like the cheeks of the bird. The pituitary gland, again, is like the little cherry hanging from their beak that they're holding in their beak. Anyway, so diencephalon includes the hypothalamus, the thalamus, and the pineal gland. It surrounds this space. Like I said, it looks like the bird cheek there. The third ventricle. It's a very thin space, but it's a space. And it's a space filled with cerebrospinal fluid. Anyway, the hypothalamus is another integrating center. This is the area that regulates sleep, hunger, body temperature, water balance, and it controls the pituitary gland, and it's a link between the nervous system and the endocrine system. Basically, the pituitary gland, you've probably heard people call the master gland. You know, it is a gland that has one half that's pure neuron and the other that's a mixture of neuron and endocrine. But the hormones that it releases control other glands in the body, and we'll get into that, but not right now, not in nervous. All right, so the diencephalon has the hypothalamus, the thalamus, the pineal gland. So the thalamus is two masses of gray matter. All the senses go through there except your sense of smell, and they are sent on from there to the appropriate spot. Like the optic stuff would be processed back here, and yeah. The pineal gland makes melatonin, which helps give us our circadian rhythm. All right, so let's move on. We're now going to talk about the cerebellum. It's that little round thing at the back, okay, on each side, right by the brainstem. And even though it's not something that we're going to talk about, I'm going to mention, see these white lines here? They keep branching out. And there's actually a anatomy name for this. They call it the arbor vitae, which means the tree of life. All right, but let's move on to the cerebellum. Cerebellum is at the back under the occipital lobe, and where it says separated from the brainstem by the fourth ventricle, let me make that perfectly clear. All they're saying is here, this is the brainstem. In fact, let me jump up. That's the brainstem, okay? And that is the cerebellum. What they're saying is that this canal coming from the third ventricle, coming along, it expands into this little triangle-shaped space. That's your fourth ventricle. All they're saying is that you've got a layer, and that continues as the central canal goes down your spinal cord. All they're saying is that you got this tube with an expanded area that's between dorsally the cerebellum and ventrally the brainstem. So that's your fourth ventricle they're talking about. Anyway, moving on. Oh, they do mention the arbor vitae. Okay, that's cool. So the cerebellum has this white tree-like structure called the arbor vitae, tree of life. And over that, you have a thin layer of gray matter that has lots of folds and grooves in it. The cerebellum helps you maintain your posture, your balance, prevents our movements from being jerky. Okay, so we've talked about the in the cerebellum. We've talked about the cerebellum. We're now about to go to the three parts of the brain stem. And so we'll talk about the pons and the medulla. And what am I leaving out? I could cheat and read. Oh, midbrain, pons, and medulla. Okay. So there are three parts. Oh, I could have gone to the next slide. The brain stem has the midbrain, the pons, and the medulla. The thing I've circled right here is the midbrain. The next slide is going to be the pons, which is right here. And the medulla is right there. Actually, medulla oblongata. So the midbrain is a relay station between the cerebrum and the cerebellum. Makes sense. We got the cerebrum here, the front brain, we got our back brain here. Okay. And this is where we send signals between all this stuff and this stuff back here. All right. And actually, I shouldn't just say that. This is also down here and here. So basically, a lot of people talk about your back brain and your forebrain. All this stuff is part of the higher consciousness. And some people talk about, oh, your little lizard brain, your primitive brain. Stuff back here, we'll talk about it. But stuff like breathing happens there. So anyway, the midbrain is a relay station. It's got centers for vision, hearing, and ability to know that you're touching things. The pons, that's the little oval right there, it controls your breathing rate, working with the medulla oblongata there, and coordinates your head movements so they're not jerky, and helps with the response to visual and hearing stimuli, things you hear, things you see. The medulla oblongata, that does things like breathing, heart beating, control of the diameter of your blood vessels. All that's controlled right back here in your brain stem. Okay, well, all this is your brain stem, but this part of your brain stem, the medulla oblongata. All right. It also has centers for vomiting, sneezing, coughing, hiccuping, swallowing. All that stuff is controlled back there. It's and in the area that's above the spinal cord, so we're talking upper medulla oblongata, we have, again, nerve tracts that ascend, that carry sensory information up to the brain going through there, and nerve tracts that descend, that carry motor information from the brain out to the rest of the body. Okay. The reticular formation, okay, this is bunches of gray matter and gray matter fibers that are going through the brain stem. Remember, brain stem is right next to the spinal cord. The spinal cord has gray in the middle, white in the outside. So this is the area where we're switching everything over from the brain orientation to the spinal cord orientation. And it's the thing that can help you focus, get rid of unnecessary stimuli. General anesthetics can mess with it. If you get a really bad injury there, you might end up in a coma. So this is what we're talking about, coming right through the brainstem. We have the reticular formation, reticular area, and it's going to send fibers out through the whole dang brain. All right, that is a check your progress. That means we've gone through brain and spinal cord, well, at least partially through. We're going to stop right here, pause right here for a second, and you can see if you can answer those. If you can, move on. If not, rewind, watch again. I'll stop for a second and then I'll continue. Excellent, let's do the thing. All right, higher mental functions. We're going to talk about the limbic system and what that has to do with memory, the ability to talk, your ability to understand people talking. So here is the limbic system. It's the stuff in blue on this drawing, okay? It integrates your emotions with higher functions, reasoning, memory. Basically keeps you from just being ruled by emotions. It has parts. One part is the amygdala. That's right here. It's near the pituitary. It's a little posterior to the pituitary. There's a gap, but the amygdala right here is what lets you, keeps you from doing stupid things and dying. It gives you a sense of fear. You don't go up and scratch the bear if your amygdala is working well, because the bear doesn't want to see you. Well, at least a wild bear. Anyway, you get my idea. Don't harass the animal. Don't step out into traffic. You should be scared of that. And that's what your amygdala does. All right, your hippocampus is like, if you think of the amygdala as a head of a tadpole, the body of the tadpole going up, that's the hippocampus. And that's involved in learning and memory. You process incoming information and, oh, I should remember that. The brain's like, that's important. So let's go through types of memory. Memory is anything that lets you remember things that have happened. Learning, a little different. You can remember things from the past and use them to help guide what's going on now. And we have short-term, long-term, semantic, episodic, and skill memory. So let's go through all of these. Short-term memory is prefrontal area. We are talking, you can remember 3 to 5 things in short-term memory. Sorry, 3 to 7. And that's things like remembering a phone number that you looked up. Long-term memory is something you've memorized that you keep in your brain. Kind of like for me when I said, oh, the cribriform plate of the ethmoid bone. That's a phrase that was drilled into my head when I took anatomy. Or a ridiculous long list of words that they had us memorize in 6th grade, and I have no idea why. But it's is, are, was, were, have, has, had, be, been, being, do, doing, did, done, does, and can, may, could, would, should, will, shall. Is, are, was, were, have, has, had, be, been, being, do, doing, did, done, does, and can, may, could, would, should, will, shall. It just, it's stuck in my head. It's long-term memory. It's memorized, in this case, memorized rubbish. Anyway, so long-term memory are things you're going to keep with you for long-term. Semantic memory would be specific numbers, specific words. Episodic memory, you remember something that happened in the past. You remember who a person is, who you know. Skill memory, this takes a while to develop. People spend a lot of time learning how to catch a ball or how to lay down the perfect bead when they're welding. But you know how they say, oh, well, it's like riding a bicycle. You do it once, and it'll come back to you if you get back up. That's a skill memory. All right, so we're about to go through a fair number of the ones in this bulleted list. Let's do this. Long-term memory storage and retrieval. You've got visual memories stored in the visual association areas, sounds in the auditory association areas. Remember, that's down low in the temporal. The hippocampus is a bridge between where memories are stored and where the memories are used. Let's back up so you can see the tadpole. All right, so the hippocampus, that's the bridge between where memories are stored and where memories are used. The amygdala is responsible for fear conditioning associating danger with memory. basically responsible for you not doing dumb things that will hurt you. All right, so here we have areas of the brain that are involved in reading, okay? If you see the word, you're using your eyes. This is occipital, okay? You are interpreting the information. So you could hear the word, you could see the word, but either way, you're going to look for meaning, okay? That information is then moved to prefrontal and then motor cortex, so you can say that word. You can move the lips, move the muscles that move the lips so you can say the word. So you see the word, you get the meaning of the word, you move that information here, and then you use that information to control the movement of the muscles. All right, long-term potentiation. After we've used a connection, remember a synapse and a synaptic cleft, we have the axon terminal here, and we have the thing that is communicating with right here. That'd be the next neuron in this case. If you've spent a lot of time sending signals between these two nerves across the synaptic cleft, this one gets better at sending neurotransmitters, which means this one will be better at responding. The connection between these two information-wise is stronger. That doesn't mean that there's a physical connection. You still got a synaptic cleft, but they're better at talking to each other. Have you ever noticed that when you're thinking, kind of not paying real close attention, but your brain will jump down a well-worn path, like, oh yeah, I know about that. I've thought about that before. We think that might be related to this. They're better at sending that signal along, and that could be used for memory storage. Could be, maybe. We don't know for sure. We're thinking. All right, so left hemisphere, you saw there was Broca's area and Wegener's area. So that's verbal. It's also logical, analytical, rational. The right hemisphere, nonverbal, creative, intuitive, visuospatial. But then, of course, you have the corpus callosum connecting the two hemispheres. So there is crosstalk, there is integration. All right. And here is a check your progress for the limbic system. Go ahead and see if you can answer these. If you can, move on. If you can't, rewind it, watch it again. I'm going to pause just a sec, and then we will continue. Okay, moving on. The peripheral nervous system. We've done central, we're now doing peripheral. We're going to talk about spinal reflexes, the difference between the somatic peripheral nervous system, that skeletal muscles. There's some stuff in there that's not voluntary, but that's what you think of as voluntary. Autonomic is completely involuntary. We divide that into the sympathetic and the parasympathetic. Sympathetic is the fight or flight, and parasympathetic would be the rest and digest. So anyway, and we'll talk about how those work in terms of the nerves and how they're different. But moving on. The peripheral nervous system, we have the cranial nerves, the spinal nerves, and the ganglia, which are nerve cell bodies that are outside of the spinal column, outside of the brain. All right. So here we have a nerve tract, a spinal or a cranial nerve. And we have 12 cranial nerves. They can be referred to by Roman numeral or name. The mnemonic device that I had to remember the names was I was taught to use on old Olympus's towering top, a Finn in German, view some horses. But don't worry about it because you're not having to do the names. You're going to do them by number. All right. Anyway, the cranial nerves have, they're mixed. Some of them are just sensory. Some of them are just motor, and some of them are mixed. They have both sensory and motor neurons. They have a lot to do with what's happening in the head, the neck, the face. The one they're saying, oh, the vagus nerve has branches to the neck and the internal organs. Yes, but when we say the internal organs, it has branches going all the way down to the heart, all the way down to the rectum and the anal canal. Well, the anal canal with the anus. So yeah, that cranial nerve 10, goes everywhere. All right, here are our cranial nerves. The first one, olfactory receptors, that is smell. And from the retina of the eyes for the second one, that's the one, the optic nerve. Eye muscles, and for 3 and 4, those are to control movements of the eyes. The jaw, mouth and jaw muscles, that would be so you can chew. Taste buds, facial muscles, and glands. Again, pretty straightforward. Inner ear, that's auditory nerve. Carries the signals to the brain. Frontopharynx and pharyngeal muscles. Yeah, these are straightforward. Glosso for tongue muscles. And 10, the vagus, goes everywhere. And tell me why 12 up there. Where's my 11, 10? Okay. It's kind of weird the way they laid this out, but that's because they're switching sides. Anyway, moving on. So the spinal cords, the spinal nerves come out of the spinal cord, and the cranial nerves come out of the brain. Pretend to be shocked by that. There are 31 different spinal nerves. Remember, they come out of the intervertebral foramina, the little spaces between the processes on the vertebrae. The posterior root, I hate that term. I'm going to be calling it the dorsal root forever. The dorsal root is all sensory, and it has a ganglia, a collection of cell bodies outside of the central nervous system, outside of the spinal cord. The ventral root is all motor fibers, and just a little ways away from the spinal cord, those come together, and then you run in a combined nerve that has both sensory and motor to the organ or muscle, whatever it is. Moving on. So the somatic system, this is nervous control for the skin, the skeletal muscles, tendons. You might say, tendons, what's with that? Stretch receptors. Not all somatic actions are voluntary. Some are automatic, but most are voluntary. We do have reflex arcs. We're going to talk about reflex arcs. So you have sensory neurons entering the spinal cord, and they stimulate a bunch of interneurons. The interneurons synapse with the motor. Yeah, lots of words. It's much easier to understand if you're not freaking out about the words. So the signal will travel in the motor nerves to the effector. In this case, that we're about to show you, it's a muscle. And some of the interneurons will carry it up to the brain, so the brain knows something even happened. What we're talking about here is, oh, I've been stabbed by a pin or something horrible. The example I used earlier was you touch a stove, a hot stove. Well, that symbol, so that signal goes to the spinal cord, And without the brain ever being involved, interneurons like, we must solve this problem. And they send a signal down the motor neuron to the muscle to pull the hand away from the source of pain. And meanwhile, yes, some interneurons in ascending tracks let the brain know what's going on, but they already made that decision. Not that you care, but there was a time I was working on a car and I was welding. Um, it and I backed into a place that I had been welding a few minutes before, and obviously it's still super, super hot. I pulled away from that without ever feeling the pain at first. And I got nasty burns, but I was moving away before I even knew it had happened, before I knew there was pain. It's like, oh, and then you're, oh, ow, ow. You look at it, oh, I burned myself. Anyway, don't weld on cars in shorts. Don't wear shorts while welding. It's bad. Okay. So we talked about the somatic. That was things, most of which, not all of which, are under voluntary control, going to skeletal muscles, skin, things like that. Autonomic, this is going to smooth muscles that do like vasoconstriction, vasodilation, or in the GI tract, they move food through with the peristalsis. And it also goes to organs, glands, talking like adrenal glands, stuff like that. The autonomic nervous system is not voluntary. We do not have the ability to control this. The two systems, sympathetic and parasympathetic, work in opposite manners. I've already said this, but sympathetic is the fight or flight, and parasympathetic is the opposite, that rest and digest is the phrase that they use for that. So involuntary, they affect the organs, and they involve the use of two neurons and one ganglia for any impulse, whether it's sympathetic and it makes you better at running away, or it's parasympathetic and it makes you better at relaxing and calming down. Either one is going to be two neurons and a ganglia. Remember, ganglia is a collection of cell bodies. So here's an important table. Yes, know the information in this table. I'm going to hit the high points early. Okay, so if we look at what the effectors are, the thing that's affected by nervous stimulation, if it's stimulation through the somatic motor pathway, we're talking skeletal muscles, which are parts of somites, body segments. If it's the smooth muscle, the cardiac muscle, glands, internal organs, that stuff is always autonomic. That means it's not under voluntary control. Now, sometimes it's sympathetic and it makes you better at running from the bear, but worse at doing digestion. And sometimes it's parasympathetic, and that would make you better at digestion and not so good at running from the bear. But either way, understand autonomic, they have the same targets, smooth muscles, cardiac muscles, glands, organs, and it's not under conscious control. Now, the neurotransmitter thing is a little different. In the somatic system, they're using acetylcholine. In the autonomic system, if it is parasympathetic, if it is rest and digest, you're still using acetylcholine. But if it is, oh no, things are going bad, I gotta fight, that's norepinephrine. Different neurotransmitter, adrenaline. All right, location of the motor fiber. Where are these motor fibers? If it's in the somatic pathway, we're talking cranial nerves, spinal nerves. If it is autonomic and it is sympathetic, helping you fight, helping you run, that's going to be your thorax or your lumbar spinal nerves. If it's parasympathetic, it'll either be up brain or down low pelvis. Okay, it could be sacral spinal nerves. The number of neurons to send a message, if it's somatic, it's 1. Long axon. If it is autonomic, it's 2. Always 2. And we already did the voluntary, involuntary. So yeah, know that stuff. All right, so the sympathetic division of the autonomic nervous system, we have fibers going to a ganglia, and it says terminate in ganglia lies near the core. So this ganglia is close to the spinal cord, and preganglionic fibers, it says arise from the middle portion of the spinal cord, terminate in the ganglia. The preganglionic fiber is short, the postganglionic one is long. If we're in an emergency situation and fight or flight, those are the choices we feel like we have, that's what sympathetic is doing. It's going to do things like increase your heartbeat, dilate the airways, dilate the blood vessels. This is so you get plenty of oxygen to the brain and to the muscles. So you know how to fight, you know where to go when you're running. So you try not to fight. This is also going to slow down and inhibit the activity of your digestive tract and your urinary organs. You don't really have time to be making urine when you need to be running for your life. You don't really have time to digest that meal. You need to be running for your life. Okay, and the postganglionic axon, the second axon, it's releasing norepinephrine. Okay. And that's pretty similar to epinephrine, adrenaline. I mistakenly, inaccurately earlier said, oh, norepinephrine kind of anti-adrenaline. No, it's like adrenaline. So anyway, norepinephrine is like adrenaline. And that's used as a neurotransmitter if we are in sympathetic mode, if we're run for our lives mode. Parasympathetic, this is the feed and breathe. This is gonna slow down your heart rate, help you to relax and chill, promote the use of food. There are a few cranial nerves, but there are a lot of fibers coming out of the bottom of the spinal cord. I'd have to look, but I anticipate this probably means the cauda equina. The end of the spinal cord looks sort of like a horse's tail, and they call it the cauda equina, which literally means tail, horse. So, and in this case, it's the opposite. The parasympathetic is the opposite of the sympathetic in where the ganglia is. So in parasympathetic, the preganglionic cell, that nerve fiber is super long. And then when you get close to the organ, you have a postganglionic cell. So exact opposite of what you have in the other division. Anyway, in the sympathetic. So yeah, so sympathetic, the ganglia is close to the nerve, close to the spinal cord, and in parasympathetic, the ganglia is close to the target organ. And we already said that helps you relax. We call this the rest and digest system, and it uses acetylcholine, the same neurotransmitter that's used mostly in the somatic system. All right, here's a check your progress. I'll pause for a second. You can read this stuff over, see how you feel about it, see if you know it. If you do, we'll continue. If you don't, if you can't answer it, go ahead and rewatch and then try to answer. I'm gonna pause this for a bit and then I'll move on. Okay, so let's talk about drug use, drug therapy, drug abuse, all that jazz. We're going to talk about how drugs interact with the nervous system, whether they are stimulants, depressants, psychoactives, and some of the long-term effects, which may not be pretty. All right. So we'll start with neurotransmitters. We've talked a lot about acetylcholine, norepinephrine. Acetylcholine is necessary for memory. Norepinephrine, dreaming, walking, mood. Dopamine regulates your mood, helps you coordinate your movements. Serotonin, again, sleeping, thermoregulation, emotion. GABA inhibits the whole central nervous system. I shouldn't say the whole. Has a strong inhibitory effect on the central nervous system. All right, so neuromodulators are chemicals that block the release of a neurotransmitter or modify how neurons respond to a neurotransmitter. An example of that, well, the book is calling Substance P. I'm sure it has a real name, but it is released by sensory neurons when there's pain. Endorphins are chemicals your body releases to block the release of substance P so you don't feel pain. They're natural painkillers. So they modulate the release. All right, so let's talk about pharmaceuticals. And there are ones that change how much neurotransmitter you have at a synapse. There are ones that will decrease the activity of that neurotransmitter, not change the amount, but change how you respond to it. And there are some that chemically mimic either the neurotransmitter or a neuromodulator. We're about to go into each one of these. So one thing that you could do is increase the amount of neurotransmitter at the synapse. That's what Xanax does, Valium. They both increase GABA levels. Reducing norepinephrine and serotonin. Sorry, if you have reduced levels of norepinephrine, serotonin, that causes depression. Prozac will increase the amount that you release. Alzheimer's, progressive memory loss. You can take drugs to increase the amount of acetylcholine to strengthen connections between, in the limbic system, and I would assume that means that you're going to be strengthening the connections between those nerves. So, continuing common modes of transmission. We just went over increasing the amount of neurotransmitter. Now let's go over changing the action. Some of the antipsychotic drugs that we have for schizophrenia decrease the activity of dopamine. They make us less responsive to dopamine. Caffeine, my Mountain Dew here. Caffeine interferes with the effects of inhibitory neurotransmitters on the brain. Now, the last one here, replace or mimic a neurotransmitter, neuromodulator. A lot of opiates, things that come from opioid plants or are chemically similar to them. We're talking about codeine, heroin, morphine. They bind to endorphin receptors and act like endorphins. So they reduce pain and they give you a feeling of well-being. Dopamine is part of the brain's reward circuit. And if you activate the reward circuit, it makes you feel super good, like pleasurable activity like eating. Drug users can take opioids to the point that they stop taking care of their physical needs. I'm talking like You don't eat, you don't bathe, you do this kind of thing in favor of continued drug use. You can continue to feel wonderful, like you're doing an awesome thing. If someone is abusing at that kind of level, there will be a dependence on the drug. The people will have a craving for it, and they'll become tolerant. That means they need more of it to have an effect, and they'll go into withdrawal if they stop taking it. Their body will have very strong negative reaction. That can happen for one reason, because you may have stopped making the natural chemical that that drug is similar to. Excuse me. And we'll talk about that. So let's go through some pharmaceuticals and talk about them in general. Alcohol will be the first one. You may be like, that's not a pharmaceutical. Well, it may be legal, but that doesn't mean that it's not a drug. And when we talk about alcohol, we're talking about ethyl alcohol. Methyl or wood alcohol is much nastier and much worse for you. But alcohol is still ethyl alcohol is still a mild toxin, and your liver spends a lot of time destroying it so it doesn't do bad things to you. Anyway, so it's a depressant, and you might say, I don't feel depressed when I drink. Well, that's not necessarily what that means. It depresses your inhibitory centers, okay? And makes you feel less inhibited, more outgoing, all that. So it's a depressant. It increases GABA, and that is an inhibitor. It depends on the amount how bad it is, depends on the amount you take. It could make you feel relaxed or lower inhibitions, like I said. It could be something that'll impair your coordination, your concentration. That's why drunk drivers are so dangerous. Huge amounts could make you slur your speech, could make you vomit. If you take large amounts, there's a risk of coma or even dying of killing yourself, because it is a poison. Now, nicotine, it causes a release of epinephrine, adrenaline. And that means your blood sugar is going to spike because your body is convinced there's some bear or horrible thing that's going to chase you, and you got to run. That means that at first you're going to feel stimulated and powerful, but the blood sugar is going to drop, and you'll have depression. You might have depression or fatigue, and it'll cause you to try and get more nicotine. The stimulated neurons will be releasing dopamine, which will give you a sense of pleasure. And it's a real sense of pleasure. That's why 70% of the people who try smoking get addicted, and only 10 to 20% of those people are able to stop. Smoking is a big factor in cardiovascular disease, respiratory disease. All right, cocaine and crack, they're stimulants, powerful stimulants, but they interfere with the ability to take up dopamine at synapses. That means the dopamine stays in the synaptic cleft and continues to stimulate, and you get more brain activity. This could last 5 minutes to 30 minutes. When it stops, you're going to have fatigue, depression, irritability. Long-term use of cocaine can stop metabolic functions in cells in the brain. There we have a patient with high levels of activity in metabolic activity in the brain before they use, someone who hasn't used huge amounts of cocaine. Here's someone who has a long-term pattern of using cocaine, and their brains are just less active. Now, we're talking cocaine and crack. Crack, of course, is cocaine that's been cut, modified for freebasing, super addictive. They're going to make the brain make less dopamine because they act like dopamine, and the brain's like, oh, I don't need to make that thing because, oh, look how there's plenty of it around. It's not, it's cocaine or crack. But that's why people will go into withdrawal when they're trying to get off of it, is they're not making the amount of dopamine that they used to. If you overdose on it, heart problems, your heart could stop, your breathing could stop. Meth and ecstasy are also stimulants. They're things that people make at home. They could smoke it, they could inhale it. You'll have the feelings of euphoria, energy, alertness, and elevated mood. But when you come down hard. You could be agitated, might even possibly be violent, irritated. Meth and ecstasy, if you take a lot or do it frequently, you could have psychosis, paranoia, hallucinations. You can see the list. Heroin comes from opium poppies, and drugs that are derived from opium are called opiates. We're talking things like morphine, codeine. They can cause feelings of euphoria and happiness. You can make pain go away. That's why people take codeine. But it's easy to get addicted. How it works: heroin will bind to endorphin receptors, so your body thinks they're endorphins, painkillers. But if you keep taking it, your body will stop making endorphins because, oh, there's plenty there. Then you need to take the drug. So you have tolerance develop, and that means you need more of the drug to prevent withdrawal. Withdrawal is really bad. You get the tremors, restlessness, cramps, vomiting, and eventually you get to the point you won't feel the euphoria, and you're not making the natural dopamine, the natural endorphins that you should be. Also, because of unsafe practices with needles and things like that, there's a chance that you're going to get hepatitis or HIV. Synthetic opiates like methadone could be used to stop someone from taking heroin, but then you have to wean them off the methadone. Marijuana and K2, okay? Cannabis, marijuana, it's the plant or the THC active ingredient. Marijuana could be ingested, but it's usually smoked. Obviously, this book is not discussing at all the fact that this has been legalized in some states for medical use, in some states legalized for recreational use. What are the effects? Mild euphoria, alterations in vision and judgment. And yes, and judgment is a real thing. Heavy use could give you hallucinations, anxiety, depression, paranoia. Synthetic marijuana. K2 is, people act like, oh, it's like marijuana, but it's much stronger, and they spray it onto stuff and use it. Here is a table, 14.2. It summarizes what we just went over, what the controlled substance is, what the effect is, and how people take it or use it or abuse it. And I would recommend that you know all of that table. Here is a check your progress, and that's the end of this video, so I'm going to stop now.